Okay, good morning, everyone. Hi, I'm Dennis Akowski. I'm the Science Director here at the Foundation. I think I know most of you in the room. Thanks for coming, and thanks for everyone who's on the webinar listening to this. Today we have a presentation by Greg O'Mullen, who's an Associate Professor, tenured at Queens College. We're very pleased to have him here today. And the topic today deals with microbes that are out there in the environment. And I find that the topic really interesting because we've all been interested, particularly those of us in urban areas, in microbes in the harbor and actually throughout the city for some time. And since the later 1800s, when there was a connection made between microbes and disease, there's been a very interesting explosion of work and epidemiology and so forth. And as it relates to water issues here in the harbor, actually it was about almost exactly 110 years ago in this very building that something called the Metropolitan Sewage Commission was formed. The group started measuring things in the harbor because the harbor smelled. Everybody knew it was bad. They didn't know really how bad. And some of the things that they measured were they actually took microbes. They actually took sediments and water samples back to the lab in this building and measured bacteria. It's kind of unclear what they were actually measuring, but whatever they found in their cultures, there were lots of them. And those early reports of how many microbes were in the system sort of advanced our understanding and the need to build sewage treatment plants within New York Harbor. So it's an interesting history. I love to send people back to the reports of that commission. But even with the interest in particularly pathogenic bacteria and viruses, people have done a good job, I guess, at measuring those things, but not a whole lot of people have gone in and taken a look at the ecology of those things, how they survive in sediment and water and aerosols and so forth. And there's a few unique people, such as Greg and this list of characters that are on the board that have actually gone fairly recently and looked at the ecology of these things. So it's something that kind of piqued our curiosity when we got a proposal from Greg a few years ago. And he and his students and colleagues have produced some very interesting work. So we're very pleased to hear that today. A couple of announcements. Again, this is the Edward Ames Seminar. We're very honored to have Edward Ames here in person today. We're thrilled with that. This will be the last seminar of the fall series. We're putting together our spring series of seminars, so pay attention to the website. And if any of you have just sort of arrived and didn't RSVP, that's fine. We have a sign-in sheet. We'd like to get your name and email and address so we can stay in touch with you. Greg.
Thank you so much for that nice introduction and for having me here today. I'm excited to be here. I am here representing a relatively large group. So the topic that I'm going to talk about today is touching on a number of kind of sub-studies and interactions with the monitoring programs. A lot of this has happened in my laboratory and in the laboratory of a close collaborator, Andrew Jewell, in association with this long list of people who I'll acknowledge at the end as well, but I wanted to put them up front because a lot of the work is actually conducted by these people. These are our students, postdoc, undergraduate and graduate students, both at Columbia and at Queens, as well as some people at Riverkeeper who have been really important in my research program as well as the monitoring work that has come out of our collaboration. So I'm excited to be here and to share some of this with you. So arriving in New York City about a decade ago, I became involved in monitoring programs in association with Riverkeeper. And we initiated a program in 2006 that really got up and running in 2008, which covered this area of New York City up to just north of Albany up to Troy. And we have 75 locations where we've collected data. About 5,000 samples have been processed. And this was all motivated by the question that I kept encountering when I would go down and stick a sample bottle into water. People just came up to you so quickly and would ask, how's the water, right? And that question has been such a motivating factor because there was a lot of areas where there was a limited amount of information available. But the reason I bring that up is there's a variety of issues associated with the quality of water. I'm really focused today on bacterial water quality, which is one of the components, and it is a component that has an acute risk. So in terms of people coming into contact with the water, infections that can occur is one of the main reasons that we're interested in water quality and in managing water quality. But that's linked to this measurement of Enterococcus, which is a fecal indicator bacteria. Okay? There are other components of water quality that are going to be somewhat beyond the scope of this talk, although there certainly are interactions. So I just want to make kind of the segment of water quality that I'm going to be talking about clear. This monitoring program has been such a great interaction for me, and we've learned a lot, I think, in recent years by sampling at these locations with Enterococcus being the primary measurement. There's a variety of other monitoring programs that are in place. As Dennis mentioned, currently overseen now down in the New York City area, the DEP does a great job of going out and collecting a lot of information that's used in the management of water quality. As you move north of the New York City boundary or border, there wasn't as much information, and that's where we've I think, had the largest impact. These 75 locations are in the Hudson or just into the mixing zone of tributaries. But that has expanded now into citizen science programs that go up into the watershed. We're gathering lots of information, some of it a little bit disturbing, some of it raising questions about what's going on with the quality of water in some places where people are surprised by the result. Down in New York City, at user locations, the New York City Water Trails Association monitoring program I've been involved with a little bit, and they're gathering great information around the city. So this amount of information in these monitoring programs is growing rapidly, and I think it's very useful, but also poses some really important follow-up questions that I'd like to start to get at today. I'm not going to promise to wrap any of those really up. We're going to take a step in that direction. So Enterococcus. Enterococcus is an indicator. I want to give you a little bit of background information. It is, there's lots of it in this room. We all carry it in our gut. It is not, however, one of the most abundant bugs in our gut. Okay? It is one of many genera that are found in fecal material. This is an image that I've borrowed here from another researcher of actually a mouse intestine, where it's a immunofluorescent microscopy image, right? So these yellow-green cells here are representing Enterococcus, but you can see that there's lots of other cells that are present. It's one of many fecal-associated bacteria. It is, however, ubiquitous. It's found in all of us, right? So it may not be the dominant component of our gut, but it is widely distributed, okay? And that makes it very useful. Generally less than 1% of the total microbial load, however, within your gut or in fecal material. So the idea here, right, is that food comes in, it travels through our system. As it travels through our system, there's a unique microbial community that is picked up, that is then deposited. That Enterococcus is one component of that, but it is intended to be representing a much broader group of fecal-associated microbes that I'm going to talk about a little bit today. And that then can be deposited to the environment. So both those other fecal microbes that we often don't measure and Enterococcus, which we can measure, is deposited into the environment, into an environment that already has, even if it's a pristine, clean environment, lots of bacteria there. Well, that's one of the things I want to talk to you about. Enterococcus is not all that abundant within our gut. 1% of the gut community is still a lot of bacteria, but it is also being deposited into an environment where you can think about the typical concentration of microbes in water being somewhere around 1 million cells per milliliter, okay? So even in clean water, there's lots of background microbes that these are being layered on top of. So what we're thinking about is these sewage-associated microbes being layered on top of the existing microbes, leading to polluted waterways having both those background microbes and a bunch of others that are associated with this input. That's the idea. I need to get back here to a couple of the points. So we're going to put this into the context of an EPA guideline. One of the EPA guidelines is the Beach Action Value, BAV, that we'll refer to. And this is, we could talk about a couple other guidelines, but to simplify it, I'm mostly going to talk about the Beach Action Value, which is 60 Enterococcus cells per 100 mils. So that's per 100 mils. So per mil, we're talking about less than 1 microbe. Okay? This is in a background context of 1 million microbes per mil. Okay? So at the threshold where the EPA guideline is suggesting a high concentration of these entero, to be harmful, it's still 1 in a million. Okay? Just keep that in mind as we think about it. However, there's lots of other microbes that are also being brought along for the ride. And entero is really indicating those organisms. Okay, so the idea is that it's coupled to these other sewage-associated microbes, some of which are pathogenic. Okay, it's the one that we measure, but it's supposed to be representing these others. It is intended to have a relatively short persistence. So an indicator, we ideally want it not to persist in the environment for very long, because we want it to be dynamic. Right? When it's entered into the environment, we want the concentration to be high, but a few days later, they die off, and that actually is a useful thing as an indicator because it allows us to see the dynamics associated with different inputs on short timescales. Okay? Things like rain events having an influence. If Enterococcus were to stick around for long periods of time, that becomes more complicated. So that's the assumption here, is that within the environment, within water, it's coupled to these other microbes. It has a relatively short persistence. And it's absent or an extremely low concentration within that pristine waterway. So when we see it in a sample, right, which is overrepresented in this simplistic image, it is representing these other microbes which are more common but not found in the pristine waterway. That's the idea anyway, right? So what I want to do is step back and give a little bit of an idea of the kind of conceptual framework for how we think these microbes move through the system. So I've already said food goes in, associated with the microbiome of the human population, Enterococcus being one component of that community. It's then deposited in the natural environment through either incomplete wastewater treatments, if we're thinking about viable microbes. And that's one of the points I should make, is most of the measurements of this are relying upon growing, cultivating it, right? So 24 hours later, we get a feel for what the concentration in the water sample was, brew it up. So disinfection will cause, will kill off those microbes, and we won't be able to grow them. So if it's deposited and we're able to grow it, we're talking about incomplete wastewater treatment, or in New York City, where our wastewater treatment plants really do a fabulous job, it's primarily a problem of the delivery system of these combined sewer overflows, which I'm not going to go into a lot of detail, and I assume most of you are familiar with CSOs. Right? When it rains, there are 400-ish locations around the city where a combination of stormwater and untreated sewage is released into the environment. The volume is too large for the delivery system to handle. We can't get it all to these fabulous plants, and therefore untreated wastewater is distributed around the city and in other parts of the state when it rains. So the idea is that is delivered here, and this is kind of a poor image, but this is one of the larger CSOs in the city. This is a little spillway that's an actively flowing CSO. It's not a great image because it was taken at about 5 in the morning, so we had to brighten it up a little bit. These CSOs are, they don't work with your schedule very well, right? So getting out to sample them is a little bit of a pain, but this image is showing water coming down the spillway, this CSO flow that's then mixing into Flushing Bay, and you see boats here and a pier in the background. So that is then deposited into the environment. So your gut has these microbes, they're deposited into the waterway. When they're added to the waterway, we think about transport, which includes dilution, as it moves through the system with the water, and also some component of decay, right? This indicator is not sticking around for all that long, so it's dying off at some rate. That leads to the environmental distribution. That environmental distribution allows opportunities for us then to come back into contact with that water and exposure pathways for health outcomes via contact with water, right? That's the general idea of the pathway of this microbe through the system, with a lot of interactions going on here that we're going to talk about more in detail. That has, in parallel to it, a management pathway that we can think about. And I bring this up because the inputs and the dynamics in the system are important to what we observe and what we can do about it. So we have this distribution within the environment that we sample at a variety of locations. We use those samples to infer the distribution in a broader area. That monitoring program allows notification of the public of harmful conditions, education about water quality issues, ideally forecasting, so we don't have to wait to tell you what water was like yesterday, right? So we can actually have more of a predictive understanding of how this works. Ultimately, mitigation, so we can actually cause improvements in the system in those places that problems occur, which then has feedbacks to the distribution, right? It becomes this cycle that hopefully improves over time. So this interaction of the microbe through the system, if we understand this, we can do a better job in the management pathway. That's the concept. And there's a lot of ecological interactions within the system that we can learn more about. Okay, so what I want to do today is I can't represent everything that's happened in the monitoring programs in this seminar, and I wouldn't probably be the best person to do that for all of those different programs, right? So what I'm going to do is just take a few examples of kind of big picture messages that are coming from those monitoring programs and some of the things that I think we can learn from them and next steps, additional questions that we can ask. So I'm going to have 4 points from the monitoring programs. That's as much of the monitoring data as you're going to see today. Okay, they're all going to be very big picture, and there's a lot of room for subtlety within that, which we really won't get at. So one of them is that despite the recent improvements in water quality in the Hudson River Estuary, and the DEP monitoring program, which goes back, not the DEP conducting the entire time, but going back over 100 years now, is a great data set to see that improvement. And there has been a lot, and we should be proud of it. There's been a lot of investment. That being said, there's still a large percentage of the samples that we collect that are unacceptable relative to the EPA's beach action value. So that beach action value that I referred to as 60 cells per 100 milliliters, I'm gonna compare things to today. We could choose a couple other guidelines, but that's what we're gonna use. Overall, if we look at the 5,000 or so samples that have been taken in the Hudson River main stem of the Riverkeeper program, we see about a quarter of those are unacceptable according to the BAV. The entero level is too high. Okay? If we go up into the citizen science tributary studies, it starts to look worse. Okay? So you get up into those tributaries and you see that about 3/4 of the samples that are collected would not compare favorably to the BAV. Down in New York City at the New York City Water Trails Association Citizen Science Monitoring Program, which is at user locations, it's about 50% of the samples are unacceptable. So the bottom line is, if we believe this data, there's a lot of work that still needs to be done for regional water quality. And if you don't believe the data, and even if you do, we need to take that data as a starting point to better understand some of these patterns in monitoring data so that we can do something about it. Okay? All right, so the second major point from these monitoring programs, contamination patterns vary over time. Okay? There's temporal variation at many of these locations. There's some that have chronic levels of chronic problems, but a lot of them vary over time. And weather is certainly one major controlling factor that we've got a pretty good handle on. Okay? And that's not surprising, given that a lot of the places we monitor have combined systems that are designed to overflow when it rains substantially. Rainfall increases the level of unacceptable conditions. And I'm going to show some of the data represented this way from the monitoring programs, right? The red is the percentage of those individual samples that would be over the BAV, over the US EPA guideline for the beach action value. So overall, I told you already, for that monitoring program, 23% of the data would be unfavorable in comparison to the BAV. We can break that same data up, categorize it according to weather, right? Wet versus dry. And in this case, what we're doing is 3 days and a 1/4 inch of rain is what we would call wet, and less than a 1/4 inch of rain over 3 days we would call dry. You have to choose something that does a reasonably good job. What we see is that overall, 35% of the data in wet weather is unacceptable, and this gets down to about 12% in dry weather. So there are still dry weather issues, but there's a big lever here, which is precipitation. We can ask ourselves some additional questions about this. First of all, do other fecal microbes, so these are the patterns of Enterococcus, that one indicator that we're measuring, do other microbes that indicator is supposed to be representing show similar dynamics? We can ask ourselves that, right? Do fecal microbes or other microbes of concern also increase under these weather conditions? And are CSOs the only source of FIV during rain? So we can start to think about other sources. Do they have high levels of Enterococcus? And then we can also ask, are they representing these other fecal microbes? Okay, so one step in that direction. One of the things I'd like to do here today is say thank you to the Hudson River Foundation, because a lot of the people who have done this work have been supported by the Hudson River Foundation in a variety of ways. One of those people is Suzanne Young, who's a two-time Kogar Fellow. She's about to finish a PhD. She's probably about a year out down at the University of South Florida now after doing a master's in my lab, doing work in a great water quality lab back there, and I think she has substantial interest in coming back this way. So there's a whole bunch of people in here who benefited from those programs, so you don't get the opportunity all that often to say thank you, so I should take it now. Suzanne Young did some work as a master's student to look at antibiotic-resistant bacteria. Now, this is antibiotic resistance very broadly defined, okay? What I'm talking about here is taking Petri dishes, taking water samples, spreading those microbes on defined media, okay, and then on some of the plates adding high concentrations of antibiotics, and when you add those antibiotics, most of the bacteria don't grow. Small portions of bacteria do grow in the presence of those high concentrations of antibiotics, and that's what we're referring to as antibiotic-resistant bacteria, okay? There are clinical definitions that can be a little more complicated, but we can then take those individuals that grow on these Petri dishes, pick them, and characterize them with molecular genetic approaches, et cetera. So it's a pretty useful approach, but I just want you to have an understanding of what it is that we're doing. So what we see here is, these are measurements from Flushing Bay. Suzanne did this a number of locations throughout the Hudson, but I'm showing you Flushing Bay is kind of our backyard laboratory, right? Higher frequency data tends to be there. So we have the level of Enterococcus here per 100 mils, and we have the level of antibiotic-resistant bacteria per mil. So right away, this is there's about 2 orders of magnitude more antibiotic-resistant bacteria than Enterococcus, but these are representing a broad taxonomic group of organisms and variable potentially across these different concentrations. But the take-home message is that they're correlated. When Enterococcus is high, antibiotic resistance is high in these temporal samplings. We can do the same thing, right? Categorize that data, wet versus dry. And we see for Enterococcus up the top, that there's about a 2 order of magnitude increase under wet weather conditions in Flushing Bay. The same thing happens for tetracycline resistance and ampicillin resistance, right? They have similar responses to wet weather. We can then take those antibiotic-resistant isolates and use 16S rRNA sequencing to identify who they are, right? And we've done that with a subset of these microbes. And another line of evidence to say that they're behaving like Enterococcus is that following wet weather, a higher proportion of these isolates are from enteric groups, right? So we see there's a background level, wet weather event occurs, discharges occur, there's a 2 order of magnitude increase, so additional antibiotic-resistant bugs in the system, and there's a higher proportion of those antibiotic-resistant bugs that appear like they're from a gut or the gut-like environment, okay? The final step in maybe connecting this to the source is, in a limited number of times, we can actually go and sample CSO flow directly. So from the correlation and from the wet-dry patterns, we're inferring an enteric source, right, with the taxonomy, and we're inferring that it's coming from sewage discharges from CSOs. But we can actually measure the CSO as well. This is somewhat difficult in the city, but in Flushing Bay, there are some places where you can do it. So we can take the water that's coming out of this spillway, what we see here is, right, turbidity and salinity and the amount of rainfall and time over the course of a number of hours. The rainfall, so we start sampling here before it's light out, and the rainfall increases somewhere around, the 0.3 inches of cumulative rainfall we get the CSO to trigger. It starts flowing rapidly. This also is complicated catching this depending upon the tidal state. But in this case, we're able to sample the CSO discharge directly before it enters the system, or as it's entering the system. And when that CSO discharge occurs, you see turbidity goes way up and salinity comes down. Okay, so we're sampling over here CSO discharge. We're now showing that same plot, but for different data, now 3 antibiotic-resistant bugs and Enterococcus, so tetracycline, ampicillin, and sulfamethoxazole, and they're generally correlated with one another. But what's most interesting about this is this cluster of data up here corresponds to the samples that are taken during active CSO discharge. So during active CSO discharge, there's a couple order of magnitude increase in both Enterococcus and antibiotic resistance, which is already above an elevated level compared to what you would see in most other parts of the harbor, right? This zone, which has that kind of frequent mixing of CSOs, already has an elevated level, but when you're actually sampling the CSO, it's a couple order of magnitude even higher than that. So this is showing pretty clearly that these microbes are linked to Enterococcus. They have the same source. They're behaving the same way. Now, I don't think we should go out and measure these things all the time, but it's showing some of the value in Enterococcus. It's behaving the way that we think it should, okay? So one of the problems here is that I want to start adding some complexity to this system. One of the problems is that fecal microbes are not actually delivered directly to the waters of the estuary, okay? Instead, what happens is, as we all know, the fecal material is flushed, it goes through a series of pipes, and then it is deposited within the system. It turns out that's actually really important to understand. Not so surprisingly, there are other microbes in this system, and it does not happen immediately. There is time for growth and decay. So there is mixing of sources. That fecal material with Enterococcus and fecal-associated microbes mixes together with other bugs that are from that urban infrastructure. Other water in the system, things that are growing along the pipes, right? It mixes together, and that's actually what I'm referring to when I say sewage-associated microbes, right? So it's the indicators, fecal microbes, structure bugs, that is the sewage-associated microbes. So what actually goes into the environment, our kind of concept of, oh, it's gut microbes that are being delivered to the waterway, well, not quite. It's gut microbes that go through a successional process and are delivered with a bunch of other microbes to the environment. But not all of these microbes coming out of CSO actually are found in fecal material. As a matter of fact, the dominant bugs that are in here are not in the gut, okay? So anyway, so when we start thinking about pristine versus polluted, we need to take that into account. And there's some advantages to doing that. As I hope to explain. So this is a slide which, some of my slides have a bit too much text, but I realize that some of them may be seen by people who are not in this room, and maybe that'll make it easier. There was a study a couple of years ago. There's a number of studies you could point to, but one of them is by Oren Shanks, who's at the US EPA, and a lot of this work is coming out of the lab of Sandra McClellan out of University of Wisconsin at Milwaukee. A fabulous laboratory group is doing just great work. And they started to look at that fecal community and the sewage community using these high-throughput next-generation sequencing approaches. So we can now characterize microbial communities in ways we couldn't a few years ago, right? Much deeper coverage. So what they did, what was unique about this paper, is they looked at, I believe, 13 different wastewater treatment systems around the country. So they took samples geographically distributed, a relatively large number of them. They also looked at samples from the human gut, from human fecal material, and from animal fecal material. Okay? And what they did was they come up with what is a fecal core and a sewage infrastructure core. So the way to imagine this is not saying it's all of the microbes that are fecal or all of the microbes that are sewage associated, but they come up with this large list of the things that are in different samples of fecal material, for example. Then you can think about a Venn diagram, right? So they're overlapping those distributions, and the things that are shared in all of those samples and are abundant become part of this fecal core, which is intended to be found widely distributed in fecal material in high abundance. Right? It's not everything, but it's a representative group, a number of taxa that can be used to represent those signals. So this leads to this microbial signature approach, which is used commonly in Sandra McClellan's lab. These two papers are coming out of her research lab. They have the sewage infrastructure core is represented generally by 4 genera. okay, and 7 families of bacteria. And really, it's 10 or so genera that are dominant in this infrastructure core. But we can come up with a list of genera that are supposed to represent the gut and fecal material, and a list of genera that are the infrastructure, but that are not in the gut in high abundance. That make sense? Okay. So that approach, we're then going to use a little bit. And if we do that in Flushing Bay, this is, we don't have we have a lot more data that is not fully analyzed, but this is kind of an initial look at some of that data with more data on the way. But this is the fecal core. So we can use high throughput sequencing of 16S rRNA from a variety of samples in Flushing Bay, right? And we can look at what percentage of the sequences that come out of that high throughput sequencing approach are represented taxonomically by that fecal core, okay? And we can compare that in samples to what was the concentration of Enterococcus that we measured with cultivation-based approaches in the sample that the sequencing was done on, okay? And what we see is they're positively correlated, which is a good thing. This is, again, an indication that the entero is giving us information about the broader group of fecal microbes, supporting the idea that this approach has some real value. It's working the way we think it should, okay? And just as an example from a couple of discharge samples, we can take about 22,000. We have literally millions of sequences now, but from individual CSO samples, we can take about 20,000 samples. We'll see that about 27% of those are from the taxa that are this fecal core. There certainly are other fecal microbes, but this is the group that we're using to represent that fecal community. And about 33% of them are representing the infrastructure core, so not in fecal material, but they are abundant within the infrastructure. So taken together, we're now tracking the majority of the microbes in that CSO flow, which is very different than looking at one taxa that's not all that abundant, that we have to use cultivation-based approaches, right? In contrast, in these same samples, Enterococcus, which we grow, right, cultivation-based indicator, it only represents about 0.04% of these CSO sequences. That shouldn't be all that surprising, because if it starts in the gut somewhere around 1%, then it mixes with this other source water. That's ballpark where you might expect it would be. But the bottom line is, it's relatively rare, and we can look at things that are much more abundant, and there's some advantages to doing that. And we can look at the connection between this and the Enterococcus patterns, because Enterococcus is easy and cheap to measure, okay? This is harder, but we can do it on a subset of samples. Okay, so the bottom line is they are generally correlated. Now, we can take a bunch of samples and group them together and look at a comparison of, I'm going to show you 2 of these plots. So this is the percent representation of the fecal core based upon these high throughput DNA sequencing approaches. And then we have the percent representation of that urban infrastructure core. So right up here is the proportion of sequences that are fecal, proportion of sequences that are from the urban infrastructure. Now, I have 4 groups of samples that I want to explain to you. The first one is what I'm labeling here kind of simply as the no entero ref. So what is that? These are a group of reference samples where we went out as part of the riverkeeper monitoring program. We grabbed a whole variety of samples, and we take DNA samples as we're doing the cultivation-based work. And these are representing mid-channel samples. So the Battery, 79th Street mid-channel, George Washington Bridge mid-channel, Stony Point, missed one, Tappan Zee mid, Stony Point mid, Poughkeepsie mid, where the intake is for Poughkeepsie drinking water, okay? So at those locations, we have a whole variety of samples, and now I've only selected samples where Enterococcus is seen as below detection. So this is as clean as these water samples get, okay? So this is intended to represent kind of our pristine local water. Doesn't mean there's no fecal impact, but what it means is it's below the detection of what the EPA would say, hey, everything's good here. We have good water quality at these mid-channel locations. So that's used as a reference, okay, in both of these boxes in white. Now we have the box plots associated with, these are the same location. This is a Flushing Bay user point. This is the public dock in Flushing Bay. And I've divided the samples out for those that were sampled during dry weather and those that are sampled during wet weather. Okay, same location, different condition, different weather condition. And then we have samples that are taken at the CSO discharge, which is, maybe a half mile or so from the dock location. okay? But the discharge samples are when the CSO is actively discharging, or, and actually the difference is bigger if I kept that statement that way, only when it's actively discharging. I've also included a few samples here at the CSO when the CSO has just discharged very recently, so we're sampling in that mixing zone. And that brings these levels down a little bit, but they're still very elevated. Now this is also worth pointing out, right? We've got a log axis here, so 1%, 10%, 100%, okay? So if I didn't do that, these are invisible, okay? Because they're all below 1%. This is a linear axis. These would just be, down here along the 0 point. Okay, so what we have is these different comparisons, and we can look at the fecal representation. There are some fecal microbes, and there's two samples that are up actually around the 1% mark, even though there was no entero. But they're very rare, right? The vast majority are below, the median is below 0.1% of the sequences that we get, tens of thousands of sequences, are falling into taxonomic groups that we put into this fecal core. Okay, so very low fecal representation. We wouldn't necessarily expect these to always be 0, okay? But that is a very low concentration. Similarly, the dry weather condition in Flushing Bay, we often see entero concentrations in Flushing Bay during dry weather that are acceptable, right? The water quality is really dynamic on short time scales, okay? The median is higher than those reference samples, but the distribution is statistically overlapping. Okay, so we have a group here, which is dry weather and our reference, no entero reference. And then we have the higher concentrations of these fecal core. When it rains, we have a significantly higher concentration of these fecal core associated with the sequences that we obtain, and an even higher concentration in terms of the median in the CSO discharge. But these actually are overlapping in their distribution. The maximum that we see in CSO is about 27%, the wet weather maximum is about 10%, and the dry weather maximum is about 0.5%. Okay, so we have these different levels of fecal representation in these samples over time, over time and space. The urban infrastructure is giving us a slightly different pattern, which is interesting. First of all, the dry weather Flushing Bay has slightly more influence of the urban infrastructure than it does of fecal. That may not be shocking even because of the way that we classify these things. Dry is less than 0.25 inches of rain over 3 days, right? But that doesn't mean there's no stormwater that hasn't entered the system. So we do actually see in what we're categorizing as dry a little bit of a broader signal associated with the infrastructure bugs, but these infrastructure bugs are not matched to a high concentration of fecal bugs, okay? Whereas we see a higher concentration in wet weather of both infrastructure bugs and a higher concentration of fecal bugs, okay? So this is, again, telling us that this kind of works, and we can use this tool to better understand some other systems, as I think I'll make more clear in just a moment. But I want to add some more complexity first, okay? So fecal microbes going into urban infrastructure adding other microbes that actually we can track, that becomes a tool for us. The sewage input goes into the system primarily through CSOs during rainfall. However, that's not the only source, right? We can make this more complicated. Stormwater. Stormwater is a complicated one. Wildlife, okay? Wildlife can be directly deposited, or it can be caught up in stormwater and then directly deposited, or these things can all combine together, go into a CSO, and be delivered to the system. So there's complex interactions, okay? But we need to think about what is the input of these if we're going to start understanding our monitoring data. So one question is entero found in these other sources, or is entero specific to CSOs and sewage? Right? And if entero is found, is it coupled to these other microbial groups of concern? Do we see a fecal signal coupled to that entero signal from these different sources? I'm about to make your life a little more complicated. Okay? So, Enterococcus. One thing we can say is stormwater is a large signal for Enterococcus. We think about this for a moment. So these are our samples that were taken. This is Angel Montero, who was a Hogarth fellow. Angel Montero in a Queen Street. Where we're sampling here is water before it goes into a pipe. Okay? So this is just interacting with the urban surface along the side of the street, not in puddled water, but in flowing water during active rain. Okay? We can actually take rain, no entero signal. It interacts with a small segment of pavement, and we start seeing that entero signal pick up. Okay? That's complicated. The levels that we see in these 8 sites throughout Queens, sampled on multiple dates, all of these sites are being delivered either to Flushing Creek or Flushing Bay. All of these have more than an order of magnitude higher signal than the beach action value. So from a management perspective, think about that, right? How many people are swimming in Flushing Bay? Well, how many people today are going to walk through streets that rains a little more heavily and there's all these puddles? What's the public health significance of that? Okay? Talk about an unmanaged component. Maybe, maybe not. Certainly important, but what's the significance of this? It is significant because that rainwater is delivered to waterways that we're trying to manage. And I'll tell you that this can be a source, because it's higher than the beach action value, it's higher than the Flushing Bay dry value, right? It's also higher than the Flushing Bay wet value for the indicator. So this is a legitimate source. If you are adding this during wet weather, you can raise the level in Flushing Bay, all right? Now, we can take those same samples, or the same approach. Again, fecal core on top, oh, this is improperly labeled, I apologize. This is supposed to read, the data is the infrastructure core, okay? So that is the infrastructure core, this is the fecal core, just like the last slide, okay? One thing that I can tell you is if we take the enterococcus concentration and the percent representation of the fecal core in stormwater, they are not correlated with one another. Okay, they were in the waterway, they were in CSO flow. In stormwater, they are not tracking one another. Okay, so there's high levels of the indicator, but it's not representing the fecal core in the same way it did within the sewage impacted water. That's complicated. Okay, now we can look at a few different groups here, and this is going to get to some other sources. We have the same no entero reference. We have stormwater. Okay, this is the 8 locations that we sampled on different dates. Now we need to get more. I will have by middle of the next month more of this stormwater data. This is still somewhat preliminary, a limited number of samples. But we have stormwater from the streets of Queens, and we also have some animal feces. So this is some samples that I collected, duck, goose, right along the edge of, in the Tappan Zee, for example, right up near, some of them up near Spark Hill, a variety of locations, also in association with Jen Epstein of Riverkeeper. We have some pig, we have some cow, right? This I've all lumped together, a simplification for the purposes here, into these animal feces, gut, duck, geese, duck, pig, cow, together. And what we see is, let's just start on the right-hand side of this plot. Well, the fecal core, not surprisingly, has high concentrations in the CSO flow and high concentrations in the DNA that I extract from the animal feces. Well, it better be, right? So, because that's what it is. So if this wasn't representing the fecal core well, we'd have a problem. But it does. What's really interesting is, look how they become decoupled when we look at what is supposed to be labeled as the urban infrastructure. Not surprisingly, when you take the feces, you don't get those urban infrastructure bugs being in high concentration. They're well less than 1% in the feces themselves. But in the CSO flow, we have both the fecal core and the urban infrastructure core. This is working really well, right? Now, let's look on the left. We have the reference sample, and we have stormwater. Now, I can tell you, I already showed you the data from these same stormwater samples. There's a lot of entero in them. Okay, I've got to speed up a little bit. There's a lot of entero in them. However, there's not a lot. The median is actually below those reference samples. The range is similar. There's not a lot of fecal material, despite the fact that there's very high enterococcus. So what does the indicator mean in stormwater? Very active question that we need to think about more. There's some management relevance to this for sure. We do see a little bit more of a urban infrastructure. And again, this is from the city streets. If I sampled this from the stormwater pipe, that would likely go way up. I haven't done that enough to know, but I suspect that is going to go way up if you actually sample it after the pipe instead of before the pipe. That'll be interesting as well. But the bottom line is, you know, the management relevance of this is really important and confusing in some ways. So entero concentrations are high in waterways, and they may remain high in some of these environments. We don't always want to hear this, but even if those CSOs are removed, we could remove all CSO flow, and we may not get to zero enterococcus level, okay? That doesn't mean we can't do anything about it. Doesn't mean we can't understand it. but there is some complexity to be grappled with there. Okay, so long-term control plans, remove all the CSOs. Are we going to get to zero Enterococcus concentrations? Well, not unless we manage stormwater too. Okay, and in different systems, this is going to be different relative importance. Go back to Flushing Bay. Entero is in fact signaling. I want to be very clear about that. This is not a reason to discount Entero in Flushing Bay. Entero is very tightly coupled to sewage, to fecal microbes, right? Because that is the dominant source there. But there may be some other systems where we'll see some levels of Entero in the system that may not be coming from sewage. They are still able to be identified to a source, and we can manage that source, but we need to understand it, okay? The other component of this is, if we just believe Enterococcus is connected to the public health consequence, then what does that mean for you on your walk home today, right? Now, because there aren't all those other fecal-associated microbes, I think it's safe to walk home, right? So I don't think that this is a reason to say never come into contact with water in the city. There's such a high Enterococcus concentration. We can add, and if you measured it, that might actually be your conclusion. But if we take some other measurements, you can start to layer complexity on that. Say, oh, actually, Entero appears to be somewhat decoupled from those other fecal microbes in these samples. But it's something really to be understood. And to be honest, would I want to sample or would I want to swim in water that was coming off the New York City streets? No, we can still manage it. Right? There are some other concerns associated with it. But this is complicated. I'm not going to give you all the answers to that today. I'm just going to make it harder. Okay? So some initial conclusions, and then I've got some more to get to. Enterococcus and other sewage-associated bacteria are correlated with one another in urban waterways and following rain. Entero is doing a really good job there. CSOs are a large source of Entero and other sewage-associated bacteria after rainfall, but stormwater and wildlife can be as well. DNA sequencing for these community or microbial signatures can be used to assess the sewage and fecal impact within a sample and the coupling to that other cultivation-based indicator, and it can help us to untangle some of these source issues. It's not a silver bullet, but it is a very useful tool in parallel with some others. And preliminary data is suggesting that while stormwater carries a very large Entero concentration, it does not carry as many fecal bacteria. And that can still be spatially variable, right? We may go to other places and find that it's a different story. Okay, so back to the big picture, right? Back to the monitoring program. We talked a little bit about wet weather. Water quality also varies on temporal scales, or on spatial scales. We talked a little bit about wet weather and temporal scales. It also varies on spatial scales. Adjacent sites can be very different in terms of their conditions. This is just one of many examples that could be used. Again, the percent in red is the percent of samples that would exceed the BAB. And Beacon Harbor and Newburgh launched right across the river from one another, relatively close proximity, very different water quality, small patch size, right? We see that all over the place. This is related largely to inputs, but it can be more complicated than that as well, okay? So while there can be very different interactions at a small spatial scale, we also have some overarching similarities that occur. So what we see is that the estuary mid-channel, compared to the near shore where there's tribs mixing in, we have much higher concentrations where those tribs mix in, and in general, just around the near shore in areas where there's particular inputs, we see some elevated signal compared to the mid-channel. So why are mid-channel locations less impacted? Why is that? There's a variety of reasons. We can start to think about the different sources and what sources are being put in. Many of these sources are specifically entering from the edge of the river, right? But we also tend to use the river from the edge, right? We boat in it, right? But we also are engaged in a lot of activities along the shoreline. So we can go back to Flushing Bay and the spatial map. One way to understand this spatial variability. These are purple boxes representing, this is where a lot of the CSO work that I was telling you was done. These are boxes representing CSO inputs, right? These are DEP sampling locations, right? They are not right on top of each other. There is some input to the system. So if to understand the spatial variability and the difference near shore versus mid-channel, we can begin to understand where are the inputs. Well, they're in the near shore, right? And after we map those inputs, we can start to think about transport, which includes a component of decay or a component of dilution, and there's decay, as we talked about. Okay, so these start to have these plumes that create this spatial distribution that we can sample. Here's another way to look at that same thing. Now we're going below water line, right? So this is a vertical snapshot of a water column with a CSO input, okay? These are supposed to represent Enterococcus. Near the input, it's concentrated. As it transports away, it starts to dilute, it starts to spread out, and it starts to die, okay? We have this in the water column. Now I want to make this more complicated again. CSOs are delivering lots of particles. Those particles are sinking relatively quickly. Some of them are transported and then sink out slowly, but some, especially these CSOs, sink very, very quickly. In fact, Bob Chance, Fugate and Chance, about a decade ago now, studied this in Flushing Bay. And they see these large particles are entering with the CSOs. About 50% of those particles are moving in a surface plume that slowly rain out, not all that slowly actually, but others are essentially immediately deposited and move along the bottom, right along the sediment. Okay, so that's a really useful example for us. This is essentially an imaging study, right, that they did to track these particles with very, very fast sinking rates. Now, there definitely are smaller particles with slower sinking rates that we actually deal with, but CSOs are delivering some particles that are they're just going to the bottom so fast. This is important because these particles can be colonized. Okay, so the microbes don't just move with the water, they're attached to particles. And this is another student who is now out at Stony Brook about to finish up a PhD. She did work and published it in 2011 where we looked at bacteria and the fecal indicator Enterococcus in a variety of locations, 6 sites around the Hudson River Estuary. I'm showing here bacteria and Enterococcus and the percent of them that are particle associated. 3 microns or larger is the size of the particles. At 2 locations, Flushing Bay and Piermont Pier, which behave somewhat pretty similarly, right? And it represents the signal of these other locations. Enterococcus is about twice as likely to be particle associated as the background bacteria within the bay. So the sewage-associated microbes tend to be particle associated, and they are therefore going to have a sinking flux. And Andy Jewell has looked at this more, and the sinking fluxes of the particles that we're measuring are a lot slower than what was coming directly out of that CSO, but it's still relevant that there's a sinking flux that can get them out of this surface water. Some of those cells are going to be, first of all, free-living and just moving with the water. Some of them are attached to particles that are still moving pretty much with the water, sinking very slowly. But there are complicated distribution of cells that are sinking, and they can make it down to the sediment. We can also start to structure the water column. Light and UV is important to the decay. So as those particles sink out, they get into deeper waters where they die more slowly, and some of them then get deposited down in sediment. Deposition in sediment becomes really important because we can do spatial surveys of entero concentrations, and we see that they're really high in these locations where we've been sampling. Okay, so we see here Enterococcus concentration in water at a variety of sites through the Tappan Zee and some down in Flushing and around the city. And this is Enterococcus per 100 grams of sediment. So we can think about this as an equal mass comparison. And they track each other pretty well. If you do this over time, they're correlated to one another. If you do this in pairwise samples, it doesn't really work that way, which we can talk about later. But if you look at the trends over time at a location, the geomeans, for example, those are correlated. Okay, the concentrations in sediments are also generally higher, especially in the high concentrations. They can be an order of magnitude higher than what we see in water. There's a lot of these bugs viable within sediments. And sediment acts as a long-term reservoir. So if we take those sediments from these collection locations and we bring them back into the lab and we track them over time, measuring them day after day to see how quickly they die. In water, they die fast. What I'm talking about here is a 90% reduction. Another way of saying that is how long it takes to get to 10% of your initial concentration. Okay? In water, a couple days. Okay? In sediment, we're instead talking tens to hundreds of days. Okay? These 90% reduction rates are measured in sediment in weeks to months, not hours to days. Okay? And the longest persistence, we broke these into, with combustion, more or less than 2% organic concentration content of those sediments. So a crude lumping of high and low organic content, which is kind of a natural break in our data. And we see that the highest persistence, the longest time that they live, which is on a time scale of months, is occurring at low temperature in high organic content. Okay? So we can start to understand the different distributions, but they're long. One of the things this means is seasonal disinfection is a bad idea. Okay. It doesn't all stay in the sediment either. It can be resuspended. One way to show this resuspension is to actually wade out into the water. So this is a trial where we sample water, and we go out and we wade around in the water for a minute like you would if you were fishing or swimming, and your act of encountering that environment can disturb the bottom and can cause water quality to become worse. Okay? So this is the change, the delta enterococcus, the change in 10 trials where we sampled before, we go out, we wade, we sample the water again. The water in 9 out of 10 trials gets worse after you wade. And the reason for that is particles are being disturbed from the sediment, those particles have enterococcus in them, and the water quality gets worse at the scale that can push you from an acceptable concentration to an unacceptable concentration. Okay? So this then now gets more complicated. We can think about just, oh, well, it goes in the water, it's transported. We can think about decay functions. No, we need to think about particle association, sinking, deposition in sediments, resuspension. This leads to two related hypotheses. One is that sediment resuspension as a source of microbes to water is more important in the shallow water systems, and especially within tribs, low-volume systems where you have a lot of sediment and a small volume of water and potentially high velocity, you're going to get a lot of interaction with that sediment. And that sediment can have long-term persistence of these indicators. Just like we talked about a fecal core and a sewage infrastructure core, we hypothesize that there is a sediment core that we can come up with. And if we can use that with molecular approaches, we should be able to get DNA signatures that will allow the contribution of that sediment-water interaction to be quantified. And we think in the nearshore environment, and especially in tributaries, where, go back to the citizen science data, 75% of the samples are higher than the BAV, right? Maybe that's because you have a lot more sediment interaction in those systems. Okay? So our map is now much more complicated. We have more inputs, we have other sources, we have complex interactions beyond dilution decay in the system associated with transport. I'm not even talking about today, but it is, I just want to mention it for a second. Eli Duker, who was a postdoc in the lab, he has shown aerosol transport of these microbes. So these come back onto land, right? This is very complicated. It's really interesting stuff that we spend a lot of time working on in the lab. That's just added here as a bar. Those exposure pathways are now water, sediment, aerosols. This is a much more complicated interaction than that first model that we started with. And what I'm suggesting is that our understanding of the microbial pathways should make the management signal, or the management cycle, also more complicated. We should take advantage of a mechanistic understanding of how different areas respond differently and alter our management approaches. Okay, so just as we made this more complicated, we can use that understanding to manage better. So in conclusion, I appreciate your patience here. Indicator organisms travel through a system. As they do, they become coupled or decoupled from microbes associated with various components of that system. For example, sewage, sediment, etc. Understanding that microbial composition can allow various components of the constructed and natural system. So if we understand the microbial composition that comes with the constructed and natural parts of the system, we can begin to untangle the complex ecology of those indicators. We should still make the measurements, but we need to do so in an informed manner, right? And a better understanding of these interactions can allow for optimization of management. Entero is clearly a very useful tool. We should keep measuring it. It's providing a very useful framework to understand bacterial water quality. But the addition of other tools like these DNA signatures and also quantitative PCR source tracking, they can help us to interpret that data. I'm not saying it replaces Entero. I'm not saying we should do it on the schedule that we do Entero, but we can use that information to interpret the really useful framework that emerges from monitoring programs. And then we can better understand the dynamics and better mitigate the sources. Okay? And then the final point here is that wastewater treatment and CSOs are not the only inputs of these indicator bacteria or other bacterial contaminants. Other sources like stormwater and wildlife, we need to explore them more. We need to understand them better. We need to understand also connections in the system, like sediment and water, better than we do right now, because it will allow us to better monitor the system. So with that, I want to thank the Hudson River Foundation, Wallace Foundation, Princeton Foundation, Sloan Foundation, and Equity Foundation for support. I want to thank all of the people who have actually done a lot of this work, and I'd be happy to take any questions. I guess so.
It's only 2 minutes. I really appreciate this. Hi, my name is Tom Sott, and I live here in the Battery Park City area. And I just...
Do you have a question?
I have a question and solution for this.
Okay.
I applied for a patent.
Could we put you off so maybe we answer some questions? Looks like you're...
It'll take only one.
Why don't we deal with some questions when you're talking? I'd be happy to talk, and some other people may be interested as well. Yeah.
I'm assuming that not all sediments are created equal. I'm kind of guessing that
Not all sediments are created equal?
created equal.
Yes.
That
True.
urban sediments would have a higher organic content, and rural might have a higher clay content. And that might change a lot of what your biological understanding of what happens to sediments.
Without a doubt, and we have shown some of that. What I'm showing here is a little small component of what we've done, and there's a lot more to be done. But yes, so not all sediments are created equally. Organic content is certainly one of the components that leads to persistence, right? The surface area interactions can be very different. The settling rates can be very different. So those are going to segregate as they move away from a source, and that can lead to microbial segregation. So absolutely, understanding those dynamics in more detail, I think, is really important. So we can think about sediment types that are most important.
You're going to discuss the flushing of these sewers during dry weather and what impact it would have on the population of bacteria.
Well, there is definitely this concept, which has been pretty well studied, of this first flush dynamic, right? And that means during these dry weather periods, you get material that accumulates. And then when flow starts through the pipe, it mobilizes that material, and your concentrations, we showed this in Alley Creek with some work, that first flush creates this higher particle load and higher microbial load. So flushing of those pipes definitely does mobilize material that accumulates over time. The more frequent those events are, the less the intensity of that would be. Are we going to remove all the microbes from the system? No. But there definitely are changes in the intensity or the concentration of what is emitted, especially early in events. So that relates also to CSOs, right? You think about different size rain events. You very different dynamics from a big storm and a small storm. This relates to Sandy. Sandy water column concentrations were not all that high, right? And the reason for that is lots of water, right? Small CSO events, you get just enough to start to trigger it, not as much dilution that occurs, higher concentrations locally. Big rain events, your concentrations, your total loading is huge, but your concentrations are smaller, you have more of a regional impact. So there's, a lot that we can understand, that we do understand, and some management associated with how we treat those pipes could be addressed in places.
Greg, we have some questions from the webinar. It looks like a couple of them relate to your animal sources.
Yeah. Is the main street source dog poop, or is pigeon a significant contributor? I'm going to be completely honest, Garrett. I don't know. So that doesn't mean I won't know or can't know, but I don't know. I think that pigeon and birds can contribute Enterococcus for sure. There have been studies that have even shown in air the impact of dog feces. So these things do move through systems. So they are all likely contributors. I have not been able to dig into source enough in that data to really understand it yet. What I know is there's not a lot of fecal signal, and the Enterococcus concentration is high in the locations, the 8 locations that I've studied, which is not perfectly representing the city as a whole. Is the main, okay, that one. Urban rodents, can DNA, RNA disentangle these and the impact of rodents? Well, yes, there are ways that can be done. Getting at species-specific differences in fecal material is slightly more complicated, but certainly is done. Tends to take multiple lines of evidence. There are historical data from the harbor before and after reductions of dog poop. What's the reduction in dog poop? More tickets? Is that what that's related to? Okay, Cooper-Scooper law. I don't have a dog. I should know that, right? That's very useful for me. I have a daughter, not a dog, and so I know poop, but not dog poop. Is there a difference before and after that? I don't know, and it would be very difficult to disentangle. I think relative to the scale of the CSO problem in the city, the answer to that is probably not. If we were to improve water quality dramatically in the city by addressing CSOs, maybe we could see that kind of a signal. What do you think about entero and overall fecal core being decoupled in stormwater? I think it's complicated and important. Is it due to particle dynamic differences, or is there an alternative entero source? Yes, I think entero tends to be surface associated. I think you can swab these surfaces. It's been seen in the metagenomic study in the subways. Entero is seen on those surfaces. Those are not all viable studies, but the DNA of them can be seen on surfaces in the subway. It is likely different strains, and we could get at that. Just the level of taxonomic analysis that I was doing is not lending itself to that strains, but we can.
Maybe you just add to that it's normal in your gut, so it's not going to kill you. It's on the surface of stuff. It's just related to that.
Yeah, the general answer to that is yes. However, Enterococcus is also, so in general, the fact that Enterococcus is on surfaces is not a reason to stop touching surfaces. I 100% agree. Just like measuring of sewage-related or bacteria from waterways being mobilized up into aerosols is not a reason to close your window or stop breathing. It's been there, you've been encountering it. The fact that you know about it today and didn't know about it yesterday doesn't change your health. Maybe you're stressed, but not your direct infection risk. But entero can actually, it's one of the leading associated nosocomial infections, right? So there are antibiotic entero strains that can cause infection. So it's not to say that there's no risk from entero at all, but the primary health risk that we're talking about is not from entero, and yes, entero being on a surface does not mean that the surface is fundamentally dangerous.
I didn't realize that tributaries were as important as you suggest as a source. Does that suggest we ought to look at ways of treating tributaries? I assume they vary widely, so whether they're dammed or not.
So I think it creates this important input to the system. I think it creates a very complicated input to the system. Studying, especially the mid-channel of the Hudson, is a lot easier than studying a tributary, right? The tributary has all of these sediment interactions. It has even changes in velocity have the opportunity to change that sediment interaction. We have this concept of, or at least I have a concept of, a hyporeic zone, right? So water actually, as it moves through a stream, is interacting below with the sediments. It has this path that's kind of doing this a lot, right? There may be a concept here associated with particles in that near shore environment, a slosh zone, right? It's a terrible phrase, and someone probably has a phrase. But there's an area that those particles are going to be, near the sediment surface in higher concentrations. Well, tributaries, small tributaries, that whole trib may be the slosh zone, right? So it's very complicated. Stormwater inputs, very complicated, right? Because the volume of the system is smaller. So therefore, stormwater can have a bigger impact than it does on the Hudson as a whole. So we should think about them. We should understand the signal. They're connected. The watershed collects all that material and deposits it. So it wouldn't be all that surprising that there's sewage which is being delivered to the Hudson that way. But it's absolutely a really important research priority to monitor it, to see the places where it's high, and then to understand why. And it's not, I also want to make the point, it's not necessarily the citizen scientist's job to figure out why, right? They can still help us in building a framework, and then we can come in and try to better understand it.
The stormwater runoff versus wildlife sources of entero. The stormwater presumably has entero from some of the animals you mentioned. Are there other sources of entero that are not animal related from the gut of any animal?
So they can be associated with surfaces. They can be associated with some plant material. There are some strain differences. So Enterococcus fetalis and Facium are two strains that are dominant in the gut. But there are a whole variety of other strains or species and strains that are both present in our gut and some that can be distributed elsewhere. So we can use, again, molecular approaches to get at some of that a little bit. They can be associated with decaying organic matter. You know, the signals, I think, in an urban environment especially, are very clear with sewage. There can be, you know, it can be in the background, but it's a signal-to-noise ratio, right? When you get into some of these other systems and in stormwater, yeah, I think there are strains that are persisting for very long periods of time, as we demonstrated in sediment, you know, perhaps for long periods on the streets. I'm going to throw something out which I think is not at all based in data, but is an idea that I kind of talked about with Eli a little bit more broadly. Aerosol transport. We know that sewage-impacted water, aerosols are formed, it's transported onto land. We see that there's connection between the types of microbes in those systems, right? Well, guess what? There's entero in those systems. Entero can be pretty hardy in terms of a bug, right? It has a variety of pHs, it's a whole variety of things that cause it to persist pretty well in some environments, including our gut. It's transmitted in aerosols. Well, those are being deposited somewhere, right? To what extent are they being deposited and then sticking around for a while? The surface association, where's it coming from? Lots of places. Some of it may be coming from our waterway and aerosol transport back onto land.
I mean, the proximate cause versus the original, the proximate source of the entero, whether it's surface related or aerosol or whatever, versus the primary source. It's all the entero primarily coming from the gut of some animal, or are there other places such as sediment where they live and multiply and reproduce and are never associated with the gut of any animal?
So they are, as a group, largely associated with the gut, but yes, there are some entero species that likely are persisting for long periods of time, not associated with the gut of an animal.
Persisting or originating?
Quite frankly, I'm not going to go there. You know, it's tough to say. These are organisms. Have they evolved exclusively within the gut environment? I have not looked at that over long time scales and the diversity of strains that exist, and whether some of those strains have deviated out into other. I think that's likely. I also don't think that means it's a reason to discount entero as a tracer of fecal material. But yes, I think absolutely there likely are, and I think when we explore stormwater in more detail, I think that's where that's going to come to light in really important ways. Flushing Bay is probably not the place to do it because there's all of these other signals. But there are places, and the city streets would be a really interesting place to examine that, and the diversity of organisms, the strains that are in different places, which of them there in dry weather, and there are, you know, evolutionary patterns that you could look at in terms of particular strains that have different distributions, different characteristics that, you know, you may be able to get at that a little bit, but I have not yet.
Question, you said you do sampling at the mid-Hudson, the center of the river. Is that right?
Well, we do four kinds of the Riverkeeper monitoring program. I should not say we. Riverkeeper and I've helped them some in helping to start out that program with them. We sample in the mid-channel at a number of locations. We sample in the near-shore environment in the absence of known inputs. We sample in tributary mixing zones, and we sample at user locations. Those are the four classifications.
Okay, my question is, we have been doing sampling of the Hudson River at 70th Street here. You know, the pier that goes out, or I or one, I don't know what the designation is. We've been sampling for about 8 years the toxic plankton bloom possibilities. Okay? Are you looking for more citizen science along the shore that we could also do it, as long as you're taking water samples every week in the Hudson? We can help out with this program too.
So I think there are lots of people who would be very open to that conversation. Rob Buchanan, who may be online with New York City Water Trails, be a really great person to talk to. Various people like.
If you go on our website, www.hudsonriver.org, you'll notice that there's an RFP from the Harbor and Estuary program for more citizen monitoring work specifically.
Yeah, but you didn't list anything on the shore of Manhattan.
It's south of there.
The lower harbor. So thinking of expanding it along the shoreline, since there are people dumping water all the time along the shoreline.
And the US EPA's urban small water grants, there's a variety of ways you might be able to leverage some funding, but there are organizations like Riverkeeper, like New York City Water Trails, a variety of people who I think would be very interested to talk to you about that. At some level, I could as well, but quite honestly, I am not conducting those programs on a day-to-day basis. There are people, who are. I interact with those people a lot, but they're better to talk to about it than I am, I think.
Right. You probably know that New York City has a CSO notification process by which they send out information. And in doing their work, they basically use three rain sources, Guardia and the Central Park. Also, to their credit, they break it down in terms of the notification by CSO, different areas, different areas.
And they know how some of those CSOs behave differently.
Right.
Via models. Yep.
The problem I see is generally weather occurs very geographically.
Mm-hmm.
So happening in such parts of Hawaii may not be happening in Sunset Park or something. No.
Yep.
And that's one part. The other is intensity, which you brought up before. So a quarter inch over 3 days could be totally meaningless in terms of pollution.
And different CSOs are going to trigger different points.
It occurs as, sprinkle during that whole period where it occurs in 6 hours.
Yep.
So the temporal and the geographic issues, have you tried to integrate those into your differences between wet and dry in different areas?
Certainly not in
very different results if you do. And just as an aside, I tend to use weather underground, which could be much higher in terms of looking at areas. I'm asking, are you trying to integrate that more smaller breakdown in terms of density, geography?
Yes. The answer to that is broadly yes, and in detail no. It's what you say is very important, and we have looked at those kinds of trends site by site and different responses to rain. One of the things on the Riverkeeper website is they have plots that you can look at the response of individual locations that the plots are made for rainfall intensity over the duration of a set number of days, and you can change that a little bit, and the entro response. And you can look site by site, and some sites respond very differently, and that's very interesting. So we encourage people to actually learn about that response, which is not uniform. We think that's really important. I have looked a little bit, for example, again, I should not say I, Isabella Riano has looked a little bit at Flushing Bay water from an undergrad who I've worked with a little bit, has looked at Flushing Bay water from the New York Water Trails program and used LaGuardia instead of Central Park data, and to see whether that creates a little bit of a difference. And to look at various water quality. So we created a kind of simplistic model that looks at 100 different scenarios for the pattern of delivery of water. So it's not just how much water, it's how much water over how much time, right? So you can come up with 100 different scenarios and see how that responds to different entero conditions. And you can look at different sources of LaGuardia versus LaGuardia is right there for Flushing Bay. But even that, it doesn't mean the delivery system is right there, right? That goes through Queens, and even, I believe, a little bit of Brooklyn. It's complicated. I have not had time to kind of get into the details of that in a systematic way. We have looked at it. We appreciate it. We think that there's a lot of, if you want to create a model, you look at those relationships site by site. And there's a lot of information to allow you to do that. And I think it's a productive exercise for understanding the response. And it may also be a productive way of looking at classes of locations, right? You could break out the types of responses you get will not be uniform across the city, across the Hudson River Estuary, and there's lots that could be done with that with existing data. And the weather underground historical data is great.
It's also important in terms of control technologies.
Well, and that's another thing. We live in a world that has sensors for everything, right? And sensors that can be developed and that exist for actually notifying when a particular CSO flows with real information at that location. That's not hard. I unfortunately flooded my lab the other day. I have a millipore water system, and somebody took out a drainage line, and they got a bunch of water on the floor, and actually it was kind of a problem.
Did you take that?
I did not in that case, but I do things like that sometimes. And on the floor of my lab, yeah. So what did I do? I went on Amazon, and I ordered a 3-pack of water sensors for $25. And my lab now has, in case an undergrad or someone removes this drainage line when it's not flowing, now has along the back wall 3 sensors that will trigger an alarm. $25, 3 of them on Amazon, right? This is not fancy technology. There are ways that this can be done. That's easy for me to say, perhaps.
Have you studied how weight bacteria that are antibiotic resistant transmitters, this DNA that they have over to other bacteria?
Have I spent? I'm very interested in that. I think it's one of the reasons to motivate some of that work. These kinds of inputs to the environment, where it persists, how that is transmitted. I've done a little bit of work with the USGS out in Long Island after Sandy, looking at groundwater in different locations with antibiotic resistance and pharmaceuticals, and it's something I'm interested in. There's a relatively new researcher up at Bard who is an absolute expert in this area. Gabrielle, I'm blanking on his last name at the moment. I apologize if you're out there. He's a fabulous person to work on those issues, and he is now in the Hudson doing some of that. So I'm interested. I have some relevant data to it, but in terms of the transition dynamics, I'm not really the expert at that. And yeah.
The question that you comment about aerosols, when I run free kayaking programs, I'm concerned about health impacts on the water. And obviously, there's a negative aerosols forming on the water that may have some, but there's a positive being on the water to do with the land-based pollutants, like people, things like that. So in a larger context.
Wait, I'm sorry. Say that one more time.
So there's a positive to being on the water. The water acts as neutral.
Oh, for us spending time with the water. Absolutely.
Yes. So when you talk about aerosols forming on the water, having problems in them, in the larger context, you avoid a lot of the dust particles, say, that are on land. We also have data that's being collected where we'll look, and it's not as though we do a lot of sampling when the wind is coming over water. It's not as though when the wind comes in the other direction, there's no bacteria that are there. There actually are a lot of bacteria. Lots of other things besides.
Yes, absolutely. So aerosols coming off water, first of all, it's not surprising that they do. It's just interesting to think about those dynamics. It's a reason to stop throwing a lot of pathogens into our surface water. And that's, respiratory illnesses throughout the world, it's a huge issue, right? Sanitation issues in surface waters of these highly dense cities is a massive problem, and those two things are probably connected in a real way. I'm not saying they're the only source of those respiratory illnesses, but it's something to understand. It's the ecology of those organisms. Is that a reason not to spend time along the waterfront? Absolutely not. There's plenty of benefits, and there's plenty of other sources of bacteria. I'm just interested particularly in that pathway.
I was trying to make the cleanest air in New York City
is along the water.
Or on the water.
Yeah, I think that is true in many locations. I don't know that it's true in every location. And there are things to think about associated with, do you want to be going over Newtown Creek water in a kayak when they're bubbling it for aeration?
Absolutely. But the Hudson River, perhaps.
Yeah, no, absolutely. Yes, I think that there are plenty of places in the city where the air is almost certainly going to be more harmful than it is over the Hudson River. Agreed. But it would be interesting to understand. And there's great data on some of that associated with colleagues working with the DEP and the DEC at what's now called the Commoner Center, used to be the Center for Biology of Natural Systems, run this massive street-level air quality sampling program in the city that looks at other sources like how close you are to highways, and there's plenty of other causes for concern.
Monitor over the Hudson.
I am generally aware of that and interested in that interaction. That is not lost. Tracy, yes.
Thank you. So I'd like to hear you speak a little more about how we should be looking at the elevated entero count on tributaries, with taking into consideration what you told us about the presence of entero in sediment. Is that entero any kind of weakened impact in terms of human health, or the other disease-causing pathogens that it correlates with? Do they have a similar die-off rate or persistence opportunity when they are introduced to sediment? So is there any way by which we should be kind of discounting or handicapping these high counts in the rivers?
So my general response to that is no. The entero is our regulatory framework. We should trust that data, and we should use it as the framework to put attention on areas that need to be better understood and better managed. So I think that's my first response to it. My second response to it is absolutely. I don't have yet a lot of information. I'll have a little more information in the coming weeks to months associated with a little bit of work in the sediments of a bunch of tributaries and the water of a bunch of tributaries, using both cultivation and a little bit of molecular data. And that relates to a couple of those hypotheses, but they're really hypotheses at this stage. I think there are tools that we can apply, and that, especially given those patterns and all of the interest and the alarming levels at which the water is seeing the VAB, is something that's very, very important. It needs to be understood for those tributaries, but also because they are a source to the Hudson. I don't have the answer for you yet, but things that are important is I think we need to have some sediment monitoring as a component of what happens, not perhaps on the same frequency, but layered in a lot of these places to be able to better understand it. I think there is a role for these microbial signature approaches to better quantify that and to look at the linkage of some of these other microbes in the system. I do think the fact that entero is persisting for longer in sediments leads to the real possibility that there's decoupling that's occurring, right, from some of these other microbes that are associated with it. So very interesting, I think very important, lots of questions, not too many answers yet. But I think it would be a really fruitful place to put more time.
Mine is just a simple question. I often see these microbes measured in terms of fecal coliform. Do you find that to be a lot less representative of that fecal core that you're talking about?
Well, so one thing I will say is that E. coli, for example, we have done some measuring of fecal coliforms. We've done some measuring of E. coli in addition to Enterococcus. We have far more data on Enterococcus. Enterococcus is the preferred indicator across a range of salinities, and we deal with a system that is connected across a range of salinities, so that's where we put our emphasis. We have done, for example, in the sediment sampling, we have measured both E. coli and Enterococcus in those sediments. They are correlated with one another, not perfectly so, but they, if you were to use one versus the other, you're going to get the same take-home message, I think. So it's not that there's no value in them. Enterococcus is preferred for a few reasons. The extent of the coupling in those systems to the fecal core, because I don't have the answer for Enterococcus, I don't have it for E. coli or fecal coliforms either. But given the general correlation that we see in water and in sediments of those organisms, although there is in the literature, we're not the only people to have looked at these kinds of things, and in the literature there's a general concept that the persistence of entero in some of these systems, there's a paper from the UK that looked at sediment and related to what was in the water and just individual transect. Entero may persist for a little bit longer in some of these environments. That may not be a good thing. So they're complicated, interesting. I generally think, however, that the information you will get will be complementary and pretty compatible. I don't think it's going to lead you in dramatically different directions. So again, I see that as a reason that the indicators work.
Greg, we have one comment and one question. We're going to wrap up.
Okay, we have measured small tributary enterocomic coliform and osmium with HRF grants. The concentrations are huge.
Yes.
I believe that to be the case, and I think it's a valuable thing to do. What is the most valuable activity for citizen science groups to perform, as specifically as you can answer? Okay, I have very distinct thoughts about this. First of all, citizen science monitoring is great. It's great for a whole variety of reasons. It adds information. Some of these measurements, I think, can be reliably done. I think that it's great when it is nested with other sampling that can help to verify some of those patterns. The citizen scientists gathering that information, they can be out there much more often. You just have to look at the range of data. Citizens being involved in the sampling is providing a lot more information for us, and that's something that's really great. What should they be doing with that data? I think there's a whole variety of steps. The next research-based steps are often things that I don't know that citizen scientists are going to take those steps. But there's value in continued monitoring. There's value in spatial and temporal patterns within these systems. Riverkeeper was, perhaps Tracy can talk to this, is sometimes frustrated because sometimes I don't want to say things until I have a whole bunch of data, right? And I do believe that sometimes it takes quite a while to get enough data. Well, the citizen scientists are creating more data. And that helps us to answer questions. That helps create a more fleshed out framework. The next steps for them, I think, are to talk to local managers. There are a whole variety of activities that can be performed. Septic maintenance, I think, is a really big one. Things like daylighting of streams are interesting issues. There's lots of things that citizen scientists can be involved with in terms of education, in terms of better management actions in their community, in terms of keeping conversations going with managers and with local politicians. That's invaluable, right? It's just so fundamentally important. So keep doing that. Do more of that in parallel with the data that you collect. But there's also all of these other actions, so one of the concerns, and I was at a conference up at Vassar a few weeks ago, and we were talking about this issue, and it can be really frustrating for local citizen scientists to see data, and then they take certain actions to try to, do something to improve, put in a little green infrastructure. Often, the green infrastructure doesn't solve the problem entirely, right? Because this is a problem that has many, many pieces and many, many sources, and it's hard to fix it overnight. So what I would say is don't be frustrated by that. And very specifically, take those steps anyway and specifically invest in steps that have other benefits. So green space is great for water quality and it has other positive benefits. Do those kinds of things because the impact on seeing those numbers fall in the trib, it's a long process and it can be frustrating. So try to invest in actions, education actions, and actions that actually have other benefits as well. Because otherwise it's going to be a frustrating process. So I hope that's specific enough.
10 seconds. Please.
10 seconds.
We designed a machine that you can install it everywhere, homes, buildings, cities, everywhere. And it turns the sewage into pulverized dust. So in the future, when we apply this to even farms, that was a good point, thank you. Yachts, homes, buildings, they will not be producing sewage in the future. We can do that. We have the technology. And I want to share this because I just got the patent papers. I don't know where to go. And I don't know how to take this into action. And thank you. We need to take an action.
Okay, we've got, we sort of ran out of time, which is terrific. I think I'd suggest if anybody in the room wants to talk to this gentleman after we've got the room available for a few minutes. But there's a lot of interest in the subject. This was great. We ran over. which is really terrific. I want to thank everybody on the webinar for sticking with us and all of you for sticking with us, and have a happy holiday, and we'll see you in the winter. Thank you very much.
Thank you.
Hey, thanks, Bert.
Thank you so much.
I have two questions. One is, I was surprised.
Yeah, for one thing, it's good to have chart. It's one of the answers. Because we have people using all different monitoring technologies, artifacts, and things like that. So one of the answers is, choose one and try to. Why is it?